Corallivorous Nudibranchs. A single adult Phestilla sea slug can consume 64.5 square centimeters of SPS coral flesh each night, where early parasitizations are epitomized by reduced polyp extension and patchy necrosis. Their nocturnal gluttony becomes conspicuous within days, yet the culprits are challenging to pinpoint because of their eminent camouflage. Phestilla species include Phestilla lugubris, Phestilla melanobrachia, Phestilla minor, Phestilla minor 2, Phestilla panamica, Phestilla sibogae, Phestilla species 1, Phestilla species 2, and Phestilla va, which are ubiquitous in tropical and subtropical regions where they range from 7 to 38 mm in length. They are ranked within the phylum Mollusca, the class Gastropoda, the subclass Heterobranchia, the infraclass Euthynura, the subtoclass Rhynchoplura, the superorder Nudipleura, the order Nudibranchia, the suborder Cladobranchia, the superfamily Phaeonoidea, and the family Trincasiidae. Phestilla subodiosa was recognized, and Tonellia chaetopterana was reclassified as Phestilla chaetopterana, whereas Phestilla subodiosa are 4 mm and whitish cream with few dorsal projections called serrata, that facilitate metabolism and gas exchange. Hydroid-eating nudibranchs like Trinchesia sibogae, Protina peregrina, and Flabellina affinis were classified within Trincasiidae, Phascolinidae, and Flabellinidae. The family Embletonidae and genus Embletonia comprises non-Phestilla montipora eating sea slugs, and Embletonia pulchra and Embletonia gracilis are found in cold and tropical seas where the latter eats hydroids. Several slugs amass host pigments while others look like the corals they eat. For instance, Phyllodesmium rudmani is virtually indiscernible from a colony of Xenia elongata. Phestilla minor is translucent to cream with a residuum of undigested zooxanthellae that likely contribute photosynthate. Individuals are phenotypically plastic because each species manifests in a variety of colors, whereas the translucent orange bodies of Phestilla panamica have yellow-tipped gray-brown serrata, which is the only member of this genus observed in North American waters. Comparative sequence analysis of their 16 Svedberg ribosomal RNA, and the genes encoding their cytochrome oxide 1 discerns most Phestilla species, whereas disparities in larval development, egg masses, and head shape and size differentiate Phestilla lugubris from Phestilla sibogae. Most animal species cannot interbreed due to geographic separation, incompatible gametes, or conflicting mating strategies, and cryptic speciation results in those that look the same, whereas sympatric speciation creates a new species from a single regional ancestor. Phestilla nudibranchs are gynochoric where 320,000 eggs in spiral masses are laid by gravid females over 2 months. Mating is conserved throughout the phylum Mollusca and takes several hours, where genitals are located to the right and the male phallus transfers spermatozoa to the female. Feeding and non-feeding zooplanktonic Phestilla larvae called velagers undergo several transformations before environmental cues from their target and non-target corals induce their settlement, which increases their range and guarantees suitable habitat. Spiral egg masses are brooded on the backs of Phestilla melanobrachia, whereas Phestilla species 1 and Phestilla minor hatchlings may spontaneously metamorphose, which slows their dissemination, but precludes their starvation. Such species may be predisposed to switching prey, while serrata and host disparities indicate Phestilla minor 2 is cryptic. 18 to 20 hours of a Porites-derived water-soluble metabolite causes Phestilla sibogae's zooplankton to cast off their swimming organs, shells, and opercula, and attach to substrate and transmute into benthic sea slugs. The host recruitment facilitating biochemical cue induces settlement at minuscule concentrations, while their zooplanktonic phase consumes phytoplankton. Nevertheless, relentless predation is offset by depositing the equivalent of 17% of their body mass in eggs per day. Phestilla lugubris and Phestilla sibogae dine on Porites and were once considered synonyms, yet they are often cream to brown and densely swathed in a multitude of nonlinear serrata with light-colored tips. Phestilla sibogae and Phestilla minor consume distinct species of Porites, while Phestilla species 2 eats only Goniopora. Captive laboratory culture caused sympatric speciation where Porites hosts were exchanged for Goniopora and Tubostria, while epigenetic modification and phenotypic plasticity appear widespread amongst nudibranchs. Therefore, aquarium corals may be more susceptible despite extraordinary host fidelity. Phestilla melanobrachia consume Dendrophilia and Tubostria species of azooxanthellate scleractinia of the family Dendrophiliidae, and their ancestors likely fed on Porites in spite of the rapid growth of Phestilla sibogae. However, predatory wrasse and butterfly fish rarely hunt in twilight. Host proximity induces Phestilla melanobrachia's settling metamorphosis, while egg to adult takes around 60 days. Embletonia gracilis and Embletonia pulchra of the family Embletonidae are thin and cream, where the latter have few anterior serrata that increase in density to their rear, while the serrata of the former vary. Pinuphiidae and Marinidae family affiliates like Pinuphius and Phyllodesmium species feed on Porites, Octocorals, Gorgonians, sea whips and sea pens. Zoanthid-eating nudibranchs, that only eat zoanthus species of polyp are classified within Aeolidioidea or Marinidae like Phyllodesmium serratum, whereas Briarium species of star polyp are eaten by Phyllodesmium briarium, where the coral symbionts perform analogous roles in the slug. Similarly, Aeolidioidea of the genera Aeolidiopsis and Spirilla recruit algal partners when they feed upon Palithoa, while Doridomorpha gardneri of the family Doridomorphidae acquire zooxanthellae from blue coral Heliopora coerulea. A prophylactic solution of Coral RX Pro or Revive Coral Cleaner in mature seawater clears nudibranchs, yet their eggs remain impervious. Infections succumb to small examples of old woman wrasse, Thalassoma balui, and saddle wrasse, Thalassoma duperi, but their adults outgrow most systems. However, corallivorous sea slugs are challenging to bioremediate, inasmuch as other nudibranch predators also eat beneficial or ornamental species. Molluscs eat using abrasive Wagele that require silica and iron for their development and regeneration, which readily deplete in recirculating systems, therefore cease supplementation during infestations. Favorrinus mirabilis of the family Phascolinidae eat nudibranch eggs and may thus bioremediate, although these slugs will remain unavailable until exporters recognize their value. Consult the complete reef aquarist available from late summer 2023 for remedial care. reefranch.co.uk